So it's a great pleasure to be here today and talk about a little bit of a background to the work that was leading up to a Surfit project for this summer. We have the great pleasure of having Timothy Wu in our lab this summer as a Surfit student. And so what Timothy is working on, he will talk mostly in a couple of weeks or so at the later presentation. But what I would like to do today is just to give a little bit of a background to where this Surfit project came from and how it's linked to the studies that we do in our lab. I could just say that the lab is located over in the Gillespie building on the main campus, so it's just around the corner from here. The Gillespie building is a neuroscience research building. It has different neuroscience programs. The one program that we are part of is the UC Irvine Research Center, and that is a spinal cord injury and repair program. So obviously then our lab is a spinal cord injury lab, and we are interested in especially the repair of spinal cord injuries, but also the repair of nerve roots that are attached to the spinal cord. So most of the work that we have done over the past, I would say probably 10 to 20 years, have been related to the lumbosacral portion of the spinal cord. And here in this anatomical figure, you see the lower part of the spinal cord, and then these thread-like structures here are nerve roots. And those that are extending below the end of the spinal cord here are referred to as the Cauda Equina. It means horse's tail, and there is some visual resemblance of that. And these nerve roots get into trouble from time to time, and that could be from trauma, anything from car accidents, sports injuries, motorcycle accidents. Sometimes people fall from down the stairs, or maybe more commonly, more high acceleration trauma. This part of the spine is particularly vulnerable because, remember, the spine has an S shape, and anywhere where you have that change in curvature is anatomically a bit of a weak point. So the vertebra, or the bony part of the spine that are over this area, are one of those vulnerable points. So at times of trauma in particular, this portion of the spinal cord, the lowermost portion of the spinal cord, very easily get pinched or compressed or injured, and these nerve roots may be separated, they may be avulsed, they may be transected or lacerated or injured in many different ways. And when that happens, there are a lot of medical problems that continue. I mean, most of them are neurological, in that when you lose these nerve connections for the lowermost part of the spinal cord and the Cauda Equina, you typically become paralyzed in the lower part of the body, you lose sensation, but you may also develop a very difficult to treat pain. And in addition, you lose control of the bladder, bowel, and there are also associated sexual dysfunctions with these injuries. So there's really a lot of neurological functions that are affected by this part of the spinal cord. And historically, most of the treatments that researchers have been working on for the past, I would say, 15, 20 years have been related to injuries higher up in the spinal cord, like in the neck or in the thorax. So very few studies and even fewer clinical trials or clinical studies have been carried out with this part of the spinal cord in mind. And in fact, most of the clinical trials that are pertaining to the upper part of the spinal cord, they use as an exclusion criteria that if you have an injury down here, you can't participate, because this injury is also different in how it affects. We refer to it as a lower motor neuron syndrome, and what that means is that the connections between the spinal cord and the periphery and the organs, they are the ones that are primarily affected. The higher up injuries are more of the connections within the spinal cord. So it's both it's a twofold problem for these patients when they have these injuries, that they have a kind of unique injury, but also they, for various reasons, typically are excluded, and so their needs have not been attended to by the medical community to the same degree as maybe the cervical and thoracic injuries. So that's a bit of the background to why we are interested in these injuries, and also, you know, they represent about 1/5 of all spinal cord injury patients, about 20%, and I would say a lot fewer than 20% of all the studies out there pertain to this area. So it's really an underrepresented area of research. And that's why we became interested in looking at this and wanted to develop experimental models. And you may remember from the title here that talking about experimental models of the spine and nerves and injuries and so forth. So that's what we'll be discussing here. So this is a schematic drawing that was published in a paper by Dr. Harriet Chang and myself a few years ago here. This describes the lower part of the spinal cord when it comes to one of the most important functions that patients want to have repaired, and that is the autonomic functions of bladder and bowel. And so the bladder function here is quite complex when it comes to how it works. We have essentially two components. One is an autonomic component, which means that it's something we don't have willful control over. And then there is also a willful component that we call a somatic component. And so what this schematic drawing shows is that from the lower part of the spinal cord, you have different nerves that exits the spinal cord, may go to autonomic ganglia in the pelvis, and then ultimately innervate the bladder, or in this case here, a muscle that's called the sphincter muscle that is very important for control of the bladder. And there's also a sphincter muscle for the intestinal system, the anal sphincter. So those two sphincters are critically important for continence in order to have control of your bladder and bowel. And that's why they are also important to study in various ways. And in fact, Timothy's project is studying at the anal sphincter of those muscles and the electrical signals that can be evoked in different injury situations. So, what is it now? It's 10 years ago that Tao Huang, my first UCLA graduate student, published this paper with us. And this is a paper that showed our first injury and repair model of these Cauda equina or conus medullaris injuries. So what we did, we developed a scenario where the most severe type of nerve root injury that could take place, and that is an avulsion injury where the root is separated from the surface of the spinal cord. That's the worst case scenario of a root injury because it's right at the border between the spinal cord and the peripheral nervous system. But by definition, this ends up being an injury to the spinal cord because it requires growth within the spinal cord of the central nervous system in order for it to repair itself. And this is something that typically does not happen spontaneously. And that is why we had to develop the strategies to overcome those deficits. So in the early work, we were able to show that when the injury happens, it is quite a devastating injury, not just because you lose the connections, but also what happens in the spinal cord. We found in the spinal cord that over time, you lose the vast majority of the nerve cells that normally would innervate the periphery, what we call the motor neurons. degenerate and die and disappear. So, and this happens over time. So this is very important to take into consideration also when you time treatments, that the longer you wait, the more nerve tissue you're losing along the way. And that's why in these kinds of injuries, very similar to injuries after stroke, that you think of stroke like a brain attack, similar to a heart attack, that time is critical for preservation and the protection of tissues. So what we did in this model was to surgically, what we call replant nerve roots into the spinal cord to try to reconnect the peripheral parts with the spinal cord. And so in the subsequent, let's just go here, in these studies we had this surgical model. We surgically replanted these nerve roots into the spinal cord for repair purposes. And this was done in rat studies. Rats that had injuries to these nerve roots, and then we repaired these nerve roots to try to get better connections between the spinal cord and the periphery. So what we needed to do in order to assess this was to develop different outcome measures. And that's where we come back to this figure again, that when we do this surgical repair, we can actually protect nerve cells. So we have many more surviving nerve cells. So that was a very good start, that the nerve roots were able to save more nerve cells. But the question was, are they doing any good? Are they able to get any function back? That was something that of course, is the big question. It's nice to have more nerve cells, but we want them to do something useful as well. And at that point, we developed methods adapted to this injury model to record from the bladder and in rodents. And this is actually very, not just similar, it's almost identical to what would happen to any one of us in this room if you go to a urologist and have a urodynamic study. So what this study does is that it puts a catheter into the bladder. We can record pressures, and we can also record from these electrodes muscle activity, or EMG, electromyography, in electrodes that in real time goes in, and we can look at these relationships digitally on the computers. And so these are examples of how these recordings can look like. This is a bladder recording here. So this is when the bladder contracts, the pressure goes up and empties, and then there's a filling phase, and then it contracts again in this way. And also in the rat, this is an EMG recording here, and during the emptying phase, you change the activity with a larger amplitude and what's called a burst pattern firing here. And so these are just multiple cycles of these voiding or emptying phases when we're filling the bladder with saline. So these are tools that we can use to assess these recordings in the rodent model first. And so what we were next able to show was that normal recordings look similar to the previous slides, like this, but when we injure the nerve roots in this way, we get flat lines for both the pressure recordings and the EMGs. We have absolutely nothing as far as reflex responses. So the model is complete when we do this on both sides of these 4 consecutive nerve roots in the lumbosacrospinal cord. So we have no reflex activity. But when we surgically replant these like this, we actually get contractions back, reflex contractions. When the bladder is filling, we get the bladder to contract spontaneously to the filling, and with also EMG activities from the sphincters. So this was actually very, very encouraging to us that in the rat model we were able to reverse or overcome some of these problems. We were also able to show that the surgery in itself had a neuroprotective effect, that more nerve cells were surviving after the injury, Not 100%, but about twice as many as would have survived with the injury alone. And that is pretty significant if you think about the potential impact on numbers and all that. And also, the more nerve cells we were able to protect, the better the function in these types of recordings. So this is a bit of a summary figure here that shows the model with the separation of the nerve root in a worst case scenario from the surface of the spinal cord. So what happens here is that we have degeneration of the nerve fibers that go out to the peripheral organs, such as the bladder, but also any nerve fibers that would go, let's say, to the leg muscles. They would also degenerate, and the leg muscles would become weak. As I mentioned before, we have loss of neurons in the spinal cord here, those that are injured, and we also have development of pain and other symptoms from the periphery. So the nice thing with this model is that it mimics the real life clinical scenarios very well when it comes to the symptoms. You get the weakness, you get the numbness, you get the pain, you get the bladder and bowel problems and so forth. So all these human clinical issues in the syndrome that we are trying to repair can be developed in a controlled fashion here and studied, and also in this model reversed to a large degree when we do the surgical repair in this way. So this is this slide here shows the positive effect of the procedure. With the protection of nerve cells in the spinal cord, we get nerve regeneration or axonal regeneration of axons growing out and reaching the peripheral targets. And even though this is a motor injury, there is also improvement of pain in this model. So it's really an encouraging constellation of potential benefits that we are able to develop here in the rat model. So at this point, we come to a crossroad, because what we need to do is to consider the findings that we have before moving forward. And so a question is always, when do you move forward to clinical trials and patient studies? And that's a very, very difficult question to answer. And I would say that the research community, you may find a number of different opinions about when to move forward to a clinical trial, to clinical treatment in patients. And the answers may vary in part depending on which condition that you are treating. If you're treating, let's say, a devastating illness where patients are not likely to survive more than months or a few years, or whether you are considering a treatment for a condition where it's more of quality of life, but it's not a life and death issue to the same degree. And so there are a lot of these different aspects that need to be weighed in. And when it comes to spinal cord injury and spinal cord injury and repair, this is a very, very new research field. It wasn't that long ago that nobody was discussing reversal of spinal cord injury and repair and so forth. So this is a very new field, and the members of the spinal cord injury research community, as well as regulatory agencies like the FDA and others, are revising and looking and discussing and figuring out what do we need to move forward. In our model, we have a potential advantage in that when it comes to the surgical repair of the nerve roots, in its purest form, it only uses the patient's own tissues. So it's not introducing a new drug, a new hardware, or a new device, or anything of that nature. So it makes it a little bit simpler, but it's still a treatment that is invasive with potential risks and so forth. And so for us, what we have decided to do in our so to say, disease team or disease community in order to prepare ourselves for what we're hoping will be a human intervention in the future, is to introduce a step for reproducing data, but also moving it to the next level. And doing that is working with the California National Primate Research Center up at UC Davis and develop a large mammal model for this. So the purpose of that is actually threefold. One is to see, can these types of procedures, when it comes to surgeries, repairs, and so forth, be feasible when we scale it up? Is it doable to do it? And the second question is, if we can do that, are we also able to reproduce the results? And then the third aspect, which actually is a very important aspect, and that is training the surgeon. And so here we work with real neurosurgeons, real spine surgeons, because in the United States, there's not a single surgeon who has done this on a human being yet. So there is also a training part that is very important. So those are the three areas that we are addressing when we move forward to a primate model, to have the feasibility, to have the reproducibility, but also training of the clinical surgeon. And so this is a first slide showing how we, in this type of work, also adapt the whole model to human-like outcome measures. Because these are the outcome measures that we also would like to see in the humans. So that's why we're using electrical recordings like EMGs, which are done in humans. We're doing urodynamic studies that are done in humans. The animals are also participating in physical therapy in the form of treadmill locomotion. We can set up devices where they can exercise, and just like any human would go to a physical therapist after a spinal cord injury. Also we use imaging studies, MRI studies, to see anatomically what is going on in the nervous system and so forth. And but the first step, I remember when I was talking with the veterinarians at UC Davis first started this project, and because this is an anatomically very challenging area to operate. And I was asking for some advice, and I said, do you have any literature on the primate anatomy? And there was a whole group of them who were looking at me for a while. It seems like forever. And then one said, well, I think there's a book from the 1920s. And so that's where we started. But then we have since developed the knowledge based upon non-invasive imaging studies, electrodiagnostic studies, and so forth. So now we have a functional model that we can perform these studies in a clinically relevant fashion to assess the feasibility, the reproducibility, and also along the way have the training of specialized spine surgeons. So what we see here are the surgical display of the spinal cord, and here are the nerve roots in the primate. And so here we only do the lesion on one side. So these animals are actually still able to void and so forth. But thanks to technology and our ability to record both autonomic recordings and EMG recordings, we're still able to see the effects of injury and repair. But these animals are still able to move around, they're still able to go to the bathroom, so to say, and are functional in many ways. So it's a refinement of the model along the way too. And then here are MRI scans, which help us both before the surgery to identify the anatomical location, because between subjects, just like in humans, the anatomy varies a little bit. And it actually can vary more than a little bit. And so in this project here, we were able to identify where the end of the spinal cord is located, and we're able to plan our surgery accordingly, and then also after the surgery to do follow-up imaging studies to look at both completion of the surgery, confirm that we have been in the right place, working with the right roots and repairing the right roots, and also see how the roots are doing after the surgery. So it's a very powerful tool using these imaging studies. This is actually a strategy that is a bit more bioengineered in its design. And here we work with actually two groups at Johns Hopkins University in Baltimore. It's a group of actually two groups that are headed by Dr. Ahmed Hoke, a neurologist and a peripheral nerve specialist. And they have, together with a bioengineering team, developed tubes that are biodegradable. And these conduits can also be dressed up on the inside with neurotrophic factors that stimulate nerve growth. And so one of the things that are going on in our lab right now is to figure out how different types of nerve fibers have a preference liking to different types of growth factors. So autonomic fibers, or motor fibers, or sensory fibers. So we want to be able to customize these fibers to attract the right type of fibers to grow out. And this is a relatively new science also. It's just a few years old that the first revelation that the supportive tissues, what's called the Schwann cells in the peripheral nerves, actually are not all the same. Different Schwann cells secrete different growth factors depending on the nerve that they are associated with. So now we're trying to figure out the nerves that we are particularly interested in. How can we optimize the environment there? And by using these types of tubes, we will be able to bridge tissue gaps between nerve roots and spinal cord. Because after severe trauma, there's often so much injury that you cannot connect directly. So we need to have something, either a nerve graft that we can take from, for instance, the intercostal nerve, but still it's invasive surgery. You're harvesting a nerve that otherwise will be healthy, and you sacrifice something there. So by using this strategy, we hope also to be able to shorten the overall surgery time and time of anesthesia and so forth. Did you have a question?
No.
No, okay. No. So these tubes are actually quite amazing, and they have actually developed them at Johns Hopkins and shown that they can bridge tissue gaps in the peripheral nervous system, and publish that also. What we were setting out to do here was to look at, can this also bridge tissue gaps between the CNS and the PNS? And actually, I don't have that summary slide. We actually have a data slide showing that we can get axons to grow into these tissue tubes and be attracted into these tubes when we put into the tube a growth factor called GDNF, or glial-derived neurotrophic factor. And this is a gene array display showing the interactions between different types of molecules that puts the GDNF growth factor into the center. And it's pretty well established already that GDNF is good for motor nerves. That is something that we knew already, but we didn't know whether we could use these tubes and attract them out. And the short answer of that is that it's a yes when we do this in the rodent. But then the question is, can we do this also and repeat this in the non-human primate? So here we were very fortunate in obtaining a research grant from the Department of Defense. They have a spinal cord injury research program, and we were approved to scale this up to the non-human primate. And here you see an example of those types of surgeries where we first identify the nerve roots here. In the primate there are different names of the nerve roots, so they have a more lumbar nerve roots than the human does, but it's the same principle. Here we have these tubes, as you can see here, and here we are surgically putting them back into the spinal cord to bridge the tissue gap as a proof of principle. And then we're able to close up the dura or the meninges to then study these animals for long periods of time. And we're looking here at 18 months for study periods to allow enough time to have nerves regenerate and so on. So this is something that is currently ongoing, and but we're also collecting a lot of data along the way. So here, for instance, we have recently developed a way to obtain bladder contraction data from the primate. We're able to record bladder contraction during voiding phases, and also to record from the urethra sphincter, even though that can technically be a challenge because of its location and small size. But the bladder recordings can be reproducibly displayed, and also after our injury, as I mentioned before, even though these animals are able to void on their own, we can see before and after in these animals that there is a difference, which also means that we have an outcome measure to follow over time and see if our treatment can also reverse quantitatively effects on electrophysiological recordings. And so this, now we're coming a little closer to Tim's project also, because in these primates, one of several outcome measures that we're looking at are EMG recordings. And here the anal sphincter recordings have turned out to be a very good way of recording functional data from these animals. So these are animals that are slightly sedated during these recordings for immobilization, and it may take maybe 40 minutes or so to do the recordings, maybe half an hour sometimes, and they're able to just get back to their normal activities shortly thereafter, and the recordings are actually very helpful to us. So here we have a set of recordings from normal, healthy primates, where we, from a baseline, have a very quiet baseline here. So the baseline here at a very short distance here. And then what we do, we have a small glass probe, about 10 millimeter in diameter, that we just put in the anal sphincter from the outside and just kind of stretch the muscle slightly. It's not more than a fecal bolus in size, so it's a physiological stimulus. It's nothing traumatic, nothing painful. The size is physiologic. And what happens is that we see from the electrical impulse in the sphincter muscle that they get activated. And the probe is held in place for about 5 seconds, which is here, and then it's removed. But even after it's removed, we have this prolonged evoked response of EMG activity that may go on for anywhere between 1 to 3 minutes in these different subjects here. And this is something that for a neurobiologist is very, very helpful, in that we have something here that we can quantify. We can quantify the duration of these recordings. We can quantify area on the curve. We can quantify amplitudes. We can think of a number of different things that can be quantified and compared. And we can also do these recordings before and after surgery. So these are subjects that had these recordings before they were scheduled for nerve root repair, injury and repair surgery, so we can follow them over a long period of time. And these data are actually, these recordings have never been performed in the literature. At least there's nothing in the literature on this in the past, so we had to do some discovery science here as well. And here there are some of the examples of how these electromyographic recordings can be quantified and displayed. And we see that most of the activity when it comes to the amplitudes are taking place in the beginning phase of the recordings, even though they may go on. This is just for 40 seconds, but recordings may continue for a long time after that. So after the probe insertion and removal, we see what we call here the different phases that the signal is changing. And so what we are very interested in doing in developing more sophisticated ways of analyzing these signals, the signal processing here. So this is part of what Tim is doing this summer, because with the quantity of data that we are able to acquire from these animals, not just before but also after, it's a lot of material. So we need to have a way of quantifying data in a time-efficient fashion, but also accurately, and also with a sophisticated mind that we can obtain quantitative data that are interpretable and meaningful and accessible to those in the labs who are not engineers. And so also here, another approach to this is to do time frequency analysis. So this is another approach that we are hoping to develop more quantitative assessments of understanding the features of how these EMG signals work. Another more of physiologic aspect of these recordings is that the probe size may actually be important as well. We have used a standardized probe size for all animals. We're able to get signals from all of them. But if you take a single animal and start with the smallest probe of, let's say, 10 millimeters, you will get a response like this. And you will have an insertion activity, and then you hold still for 5 seconds, and then removal activity, and then you have the evoked response that keeps displaying in this fashion. So then if you go up to a higher size, bigger size, you may end up with more of a response, both in duration, but also in area under curve quantifications of these EMG spikes here. But if you go one step more, you don't necessarily get more of a response. You get up to a peak and then it declines. So there is a maximum, and this presumably has to do with stretches of the muscles. When you have contractile filaments in the muscles, if you've studied muscle biology in courses on campus here, you may have come across that there are contacts between muscle filaments like this, and if you stretch them too far, they're not able to contract and work together. So there is a maximum that you would identify as well. But again, what we are interested in right now is to develop better tools at quantifying EMG activity. Some of the basic tools are, of course, duration, maximum amplitude, the area under curve, and so forth. But also a number of other angles to this that would be very useful to explore as well. These are examples of recordings from a primate after surgery and repair. So just examples here how the kind of data that we can obtain. We can record from the left side of the muscle, from the right side of the muscle, and also bilaterally, meaning both sides at the same time. And we can compare this before surgery and then at different time points after surgery and see how these signals may change over time quantitatively. Because when we have a lesion that is one-sided only, and we have a midline organ that receives input from both sides, there could be very interesting signs of recovery and plasticity. We could have compensation from the other side in the early phases, but then when regeneration is in place from the injured side and repair, when that eventually and hopefully takes place, we will have two different kinds of mechanism, and we need to have a way of sorting out on a time scale the different contributions to the overall recovery and repair in this model. So that's why we are interested in looking at these types of recordings from many different angles to be better able to interpret EMG in a user-friendly fashion. Yes?
I do have a question now. So in the center slide, the 3-month post-surgery, is the big variation in amplitude because something else is compensating? Is that what you're saying? And then 6 months later, the nerves have regenerated, so they're doing something different?
You see, different components of the EMG signal may mean different things. You see, one nerve fiber may contact multiple muscle fibers. But if muscle fibers are denervated, that they lose innervation from other fibers, there are empty sites up there, then those that are still there can expand. So you may have what's called larger motor units, meaning a motor neuron, a nerve cell, can actually innervate more muscle fibers than before. And when that happens, you can have a change in the amplitude. So there are different properties of the EMG signals that may change as a result of how many fibers they innervate and the complexity of the connectivities and so forth. So that's why we may see also here that there is a difference between the left side and the right side. The left side is the injured side. And it makes sense that the left side may have a smaller overall signal than the right side, because the right side still has its innervation. But the left side, so here having a side difference is actually not surprising, because the left side is the injured side. And 3 months is a relatively short period of time for repair. We're looking at between 1 and 2 years to reestablish connectivity. So 3 months is rather early. So I wouldn't expect to have a lot of regeneration-induced repair at 3 months. So this is not surprising to me in that fashion. So there are a lot of different things that can be looked upon and taken into consideration. And when it comes to the EMG signals in quantifying different aspects and properties of those signals. So this is actually a couple of slides I borrowed from Tim's recent presentation to the Surfit leadership team, I assume it's called, or something similar to that. And so what we're in the midst of right now this summer is to look at EMG signals that have been obtained over the past 1.5 to 2 years from these animals, where we have animals that had the injury, animals with these nerve guidance tubes that we're using to bridge, but also animals that have had a nerve graft with an intercostal nerve. Because we want to see which approach works the best, and are there any better than no treatment, so to say. And also we want to make sure that it's not just success that we're looking for, we also need to have ways of screening for potential complications. Because when you are proposing a surgical treatment, you also want to make sure that many of the safety aspects are in place. So EMG can also help in sorting out whether responses are physiologic versus pathologic. So some of the activities that are now going on this summer is for Tim to work with a graphic user interface. And so we have different programs that we collect the data with. One is called BioPack, and then also we're using MATLAB in the lab. And so Tim is working with making it easier to interact between the different software environments and develop different displays and ways of presenting data and analyzing data in a efficient and correct way, and having different graphic displays that would be helpful for the team to use for the analysis. And And science is a team sport, and we are fortunate to have a large group of collaborators, both here at UC Irvine, but also up at UCLA and around the country and abroad, and also that we over the years have had the good fortune of being supported by outside agencies as well. So you've been very quiet, so I'm hoping you were saving any questions till the end here, so be happy to...
Thank you very much. Okay, questions for Dr. Hafton, and I asked him before if he'd be willing to answer broader sorts of questions about medical careers, and he said sure. So if you have a question about electrobiography, that's fine, or about how to get into medical careers, anything in between. Yeah, fine.
What kind of problems have you faced translating from an animal model to a human model? Have you even tried translating yet, or are you still trying to work on the non-human primates?
No, well, you mean from the animal to the human?
Right.
Yeah. So what we actually did, we created a like a functional disease team, including multiple neurosurgeons. So we actually had a conference on this a few years ago where we brought in what we consider the top academic spine surgeons from around the world. Actually, we just went down the list and invited them, and they all seemed to wanting to attend because this was such a new concept to address this. So some of the things that we're doing now in the translational aspect is a direct consequence of the dialogue that we have had at that point, but also continuously with the spine neurosurgeons, because they are the ones that eventually will be performing this procedure in humans. And I think that some may be in their own preparedness be closer than others, because I think also surgeons, they do their own risk-benefit assessments and weighing, okay, what do we know, what do we need to know, you know, how close are we, how far away are we from this treatment. And there are certain things that could be in favor of introducing this, and one is that a large number of the patients after these traumatic injuries in the acute phase will go into the operating room regardless. And this is to stabilize the spine because they have a broken spine. They may have blood clots, they may have some contamination, they may have bone fragments that could be damaging tissues further and all that. So surgeons often go in and they open up, they look and see what's there, they remove blood clots, they remove bone fragments, they clean up the wound, and then they close it up. But they don't do anything to repair things that are broken because there are no protocols for that. There are no treatments. There is no gold standard for what to do. So what they're doing is limiting further damage and hoping that decompression, if there's pressure on the nervous system, that's regarded as something that is not good. And there is literature supporting that if you remove pressure early, it may be better than waiting. So that's why nowadays many neurosurgeons are doing the spine procedures as early as they can to remove pressure early. And there are several clinical studies that have been looking at these and are still being published and so forth. So I think that for our procedure, if we can be like a piggyback upon those procedures in the acute phase, that would be a nice niche. And some surgeons are now at the point of considering this, that we may be close to that point. One of the difficulties that surgeons are facing is that if they have to open up the dura, which they would need to do our repair procedure, we cannot do it with an intact dura, you need to open up the dura. If the dura is not broken, that is one of those things that is a bit of a challenge for a surgeon to justify, shall I open a dura that is not yet broken? But if the dura already is open, they typically would explore to make sure that there are no damaged structures there. So we've actually found a whole range of thoughts and ideas from the surgeon's perspective, because they're all individuals, and they're all, especially when you invite academic surgeons who are also pursuing their own science and ideas and thinking and all that. But it's been a very, very rewarding process, and I think that we are much better off now than we were a few years ago. And I would say that for me, it's more important to get it done right than to be first on this. There are very few people out there doing the science on this right now. We're not aware of anyone doing it in large animal models right now. So we have a unique database here, and we want to make sure that the database is analyzed properly and it becomes a database for the whole scientific and clinical community. And that's why we are very open with our communication with our other clinicians and other teams.
That's,
Yes.
What are the tubes made out of, and how are they attached? And
Yeah.
would there ever be cells inside them, or is it just factors?
Yeah, so the tubes made of PCL is the polycaprolactone. It's a biodegradable. So the nice thing with these tubes is that everything that's inside the tube, every component already is FDA approved. But it's just that the tube, after it's been put together with all the components, I don't think yet it's FDA approved. I think they're still in the process of considering that process, and I'm not really sure what the status is there. But each component that it's made of individually has been FDA approved at different places. So it is something that is biodegradable, and also what they can do by changing the composition in the percentages of the different participating components, you can also let it have a longer and shorter biodegradable life. So if you have a longer stretch, you may want to have it more slowly falling apart. We have no interest in keeping this in the body forever, and so having a biodegradable, structure that's there temporarily as the nerves are crossing over. That's ideal, because in the end we just want the nerves there. We don't want this for decades. But this is something that can be there for weeks and months and so on. And they are also doing second and third generations of these tubes that are a bit smarter, so that you can have gradients of release. So you have a nerve fiber kind of growing, and they sense that it's more and more and more and more and more. So they kind of grow towards a gradient. And also so you can have gradients so you don't get trapped in there. We don't want everyone to just kind of come in and stay there. We want to come in and then go out. So there are different ways of working with gradients and components, and also having more than one factor. And that's why, in the end, I would envision that if you have a patient with a certain phenotype as far as the injuries, that you may customize this already at this level. So it's individualized healthcare at the level of the grafting. That I see as a goal that would be that could also be supported by the science, that you can actually customize these things.
Yeah. How are they attached? And would there ever be living cells?
Well, here I think that's better visible here. Actually, it's not so well visible, but here they are attached with microscopic sutures. This is all microscopic surgery. I don't know if you're it's 10O sutures. So what that means is it's incredibly tiny. It's so tiny you need to have a surgical microscope and suture these. You see that the nerve root has a sheet on the outside called epineurium, little connective tissue on the outside. And also the tube itself has a wall. So we can put a stitch in the wall and anchor it to this very thin connective tissue around the nerve root, and lock it in there. And then this end that will be inserted into the surface part of the white matter of the spinal cord, that will also have an anchoring stitch into the meninges that surround the spinal cord to hold it in place. But this is also why we, after the surgery, we like to go in with the imaging and MRIs and other things to make sure things are in place, things haven't popped out, things are looking good, and all that. So that the non-invasive imaging is phenomenal in guiding us in making sure that these things are at least anatomically in place.
Other questions? Nikita?
So earlier you showed us the schematic drawing of what's going on, and it seems like there's a lot of information and data about the different interactions between parts and components and parameters and properties. Do you use any computational models to predict how a method may play out or run simulations to sort of get preliminary findings?
It's a very good question, and we have not. And I think that there is a shortage in those models. It would be great if those models would become more available. It's actually a question that we have to answer every time that we submit an animal protocol. And I think that the way most people answer those questions is that the biological reality is more complex than the best model that is currently available. But that is today in 2013. Who knows what's going to happen in the future? Part of what we do is discovery science in learning more about the biology of the circuitries of the nervous system and all the different synaptic connections and all these things. And that is, of course, something that computational biologists are really interested in. And one of my former teachers told me that there is no simple nervous system. So even when you're studying very the invertebrates or others, it's still very complex. It gets very, very complex very quickly. And as you can imagine, in mammals, in not just humans, but also in rodents and primates, and also rodents and primates are wired quite differently, and that's another reason why we find it important, especially for spinal cord injury, to have a primate step, because in medicine there have been a number of studies that have gone from one paper, one lab, one finding to a study moving up quickly, and sometimes it works and sometimes it doesn't. But we feel that with the spinal cord injury population, it's a new research field, and in order to keep the patients and their families trust, we want to do it right. We want to go out and have, but at least we feel at the end of the day that we've done everything we can do to prepare ourselves. Now we go forward instead of rushing it. And that's why we try to work methodologically in this fashion. But if we could supplement these types of studies also with computer-based models, it would make it even stronger.
Yeah. Fine.
Very impressive work. I apologize for coming late. But there's two questions. In the case of degenerative diseases, where the connectivity of the spine, I guess most probably from the bottom up, starts losing functionality. Would this technique repair some of that?
It's a good and interesting question. I'm not sure what you mean by degenerative disease. Are you talking more about like Alzheimer's or ALS or those types of
sclerosis?
Multiple sclerosis. Okay. Yes. The practical advantage, at least to a scientist, when you work with these types of models, when you work with trauma, is that you know what happened and when it happened.
Mm-hmm.
When you are studying degenerative conditions like Alzheimer's, Parkinson's, even multiple sclerosis, when you induce that in experimental animals, you don't have the same control over what happens when.
Mm-hmm.
And so it makes it harder. I think that from an optimistic perspective, if we get this to work, I'm hopeful there would be components of this that would catch the attention also of other conditions. So that's the history of science is that way, that if something works in one disease or one condition, it will also be tested in others to see.
Are you aware of somebody else who has been looking into that?
You mean across the diseases?
Yeah, or like multiple sclerosis?
Well, multiple sclerosis has, I would say, the last 10, 15 years shifted its research field a lot. It used to be very, very dominant by immunology in trying to control the immunology lesion. Now, multiple sclerosis and the funding agencies that are supporting multiple sclerosis research are much more focusing in on repair. And in that transition, there was a very, very interesting dialogue between the multiple sclerosis research community and the trauma research communities, including the spinal cord injury communities, because there needed to be exchange of information, and that is taking place. And there is actually a fair number of research labs that are pursuing both conditions and setting up studies in both and testing because when you focus more on the repair side, that's when you need some of the knowledge of the circuitry and of the interventions. And that has now has become more and more established in the MS research community as well. And as so many other things, it was in part driven by funding agencies really wanting it that way.
If I may, a follow-up question. So trauma is, so speaking of trauma, this is addressing the spine, because of the brain. What's being done there to,
Oh, there is, it might even be a larger research community there right now, because traumatic brain injury has really received a huge federal boost in funding. Much of it is because of American troops returning from the battlefields with traumatic brain injuries of different types. And these are not just like post-traumatic stress injuries, but actually physical injuries to the brain of physical trauma, and it's a huge problem. And so there are research studies at all levels, anything from sociology, psychology, to the nuts and bolts of anatomy, physiology, repair of these injuries, and developing experimental models and outcome measures. And I think from Department of Defense and the VA and so forth, my impression is that funding opportunities are even more prevalent for the traumatic brain injury setup than it is for spinal cord injury. And that has in recent years received a lot of attention because of the unmet need that is out there right now.
And in terms of repairs, are we doing similar techniques?
Well, yes and no. In our studies here, we are looking at very much the peripheral nervous system part, not just the spinal cord part. The brain is a bit more complex in many ways, because you have so many different centra there for cognition and thinking and decision making and memories and all these things, in addition to the motor tasks. You can look at a traumatic brain injury and focus in on recovery of, let's say, motor function. But then if you only look at that, you're not taking into consideration whether someone is forgetful or indecisive and has poor attention or is depressed and so on. So it's a very, it's a complex issue. And I think that different research groups are addressing different components of this and in trying to reverse or to protect the nervous system after the trauma. Because a lot of it is rehabilitation, trying to recover after the injury. But there is also research into what's called the golden hour. If someone has a battlefield injury and his or her buddies are bringing that person back, is there something that can be administered before they even arrive at the hospital? So some of the research is how can you optimize the likelihood of a better outcome right then and there. But then you have, once someone is stabilized in the hospital and so forth, looking at, okay, now what can we do? Can we do something here like this to repair? Or can we do something pharmacologically, or can we do something maybe with physical therapy better than we have been able to do in the past? And there are a lot of different angles, and I think the long-term answer is probably a combination of things. And that's also what we are looking at. I mean, what I described today is a rather simplified, straightforward approach of how this could work. But I think in the end, it's going to be a lot more complex when we weigh in the different types of subsets of neurons, different types of nerve roots that may or may not be involved and all that. But we're on the journey of discovery science at the same time as we are also doing the translational science and moving from a small animal model to a bigger animal model, and hopefully knocking onto the door of clinical studies to have something that will be available to patients as well.
Okay, we're a few minutes past the hour, so I think we'll call a formal into this. It's probably available for questions afterwards. Dr. Hampton. There is more pizza, so